So this lecture is going to be on the cell cycle. So how cells divide, right? How that division is regulated. And importantly, when that regulation goes wrong, how can cancer arise? Because this is really where cancer stems from, is divisions of the cell. So here are your learning objectives for this lecture. Some additional resources. You could take a look at your textbook, pages 103 to 109, or you can look at the OpenStax resource, where you'll see things in chapter 3 that pertain to what we're talking about today. Another good resource is this Amoeba Sister video. Videos are always more easier to get through. So if you take a look at this video, you'll see some resources on the cell cycle, which will help explain it in an easier way. So here's kind of our what we need to get through today. So going through an introduction of what the cell cycle is, right? Why does the cell need to divide and replicate? Then we're going to look at the phases inside of it. So there's different parts of the cell cycle. There's the interphase, there's the mitotic phase where the division actually happens. And then we're going to see how it's regulated and those abnormalities that we said in cancer. So let's start with the introduction of what the cell cycle is. So there's two types of cell divisions for two different types of cells. First of all, there's these somatic cells, which are most of the cells in our body. All of them except those reproductive cells that we'll talk about. So neurons, epithelial cells, blood cells, muscle cells, they are somatic cells, and most of these will divide by mitosis. Okay, this process is where the cells make an identical copy of themselves, right? They're not making a new kind of exchanging DNA information. They're making an exact replicate of themselves when they undergo cell division. So this is very good for tissue repair, right? Your skin cells, right? Your epithelium, let's say you injure it, then your cells that are there will go and make identical copies to go replace the little bricks that got missing, right? Gametes, on the other hand, gametes is another word for reproductive cells or sex cells. And there's two kinds of sex cells. There's the egg or the ovum, right? That is a cell. And there's the sperm, right? So both of these are called gametes. They're reproductive cells. And they don't divide by mitosis. They don't make an identical copy. Instead, they fuse together through the process of meiosis, right? And they undergo a division that really kind of will make your gametes, right? So they divide by meiosis and they give rise to the sperm and the egg, and then you could have fertilization happen, right? So these have half the number of chromosomes as the original cell compared to those somatic cells that we had, okay? And that's because, think of it, when you have a mother cell and a father cell come together, you're going to end up getting kind of half of each of those pieces of DNA, right? So we're going to be focusing a lot on these somatic cells, mitosis, and how we go and we make new cells constantly in our body, right? Because we're going to want to link that to cancer. This reproductive physiology is not going to be talked about in this lecture. So first of all, why do cells divide? There's two main reasons: for growth and development. An embryo will develop in the womb, right? We have a fetus that goes and grows, and those cells need to divide and make a bigger and bigger organism. But even once that baby comes out, right, body growth happens. In adolescence, we grow, right? Those bones that grow, the skin that, you have more surface area, you need to make more cells as you grow older. And secondly, tissue renewal and repair. This is what I was talking about when let's say your skin, right? You're gonna want to, if you have an injury, you're gonna want to re-epithelialize. You're gonna want to put, let's say you have a little scratch or a scab, right? You're gonna want to make new skin cells so that you could go and fix that tissue that was damaged. Or even, right, that's repair, but renewal, some tissues are constantly renewing. So the intestine, for instance, constantly is sloughing off and remaking new cells. Your skin too. You don't need to have a scratch to make new skin cells. Your skin cells are constantly dividing. The reason why you have so much dust in your house is actually your skin cells that are constantly going and sloughing off, and you need to make new ones underneath to keep going, right? So the cell cycle describes the series of events that occur in nearly every single cell, with a few exceptions. This includes the time between the formation of the cell and when it divides into two daughter cells. Here you see this one cell divide into two separate daughter cells, right? We call them daughter cells because the one parental cell splits into two what we call daughters, right? Here you see a parental cell, right, that might undergo a division like this and form its two new daughter cells, right? You see it in the process here and here dividing into two. So the cell cycle involves three kind of big themes. So first of all, growth of the cell following division. Once a cell has divided into a daughter cell, that cell will keep growing and kind of making the proteins it's made to make, right? It's going to go and do its job and live. Then at one point, we start copying the genetic information, right? So we're going to go make a copy and get ready for cell division so that we could split that same DNA into two cells. One thing that, is important to retain is that every single cell in our body has the same DNA. It's just that maybe in the skin, you'll turn on and off certain genes of that DNA to make skin cells. And in a neuron in your brain, you're going to turn on and off certain genes of that same DNA to make things that are useful for a neuron, right? So every single cell has the same genetic information. We need to have that genetic information persist throughout our body, right? Once you copy that genetic information, then there's a process where you split the cell up, and the two cells can have now that one copy each of that DNA. Okay, so we grow. This is the big chunk of your life, right? And then once the midlife crisis hits, then they copy the genetic information and they split apart. They distribute the copies to their daughter cells. Okay? Let's go through that phase, that cell division, the cell replication, right? So the cell cycle is really two main phases. You see them very well here. There's this green over here, interphase, that consists of three parts. Interphase is about 95% of the cell's lifespan, right? It's in dividing cells, you might think this is short, but cells live about 10 hours in most dividing cells, right? So there'll be an interphase for about 10 hours. And very quickly, they'll undergo this mitotic phase, right? This M phase, where they're going to go and really quickly undergo their division. Okay, this is about 5% of their lifespan. In interphase, there's three parts to it. There's this big growth phase, G1. There's this DNA synthesis, S phase. And there's this G2 growth, second growth phase. Okay. In the mitotic phase, we'll see that there is mitosis that occurs following cytokinesis in a very short period. So in the mitotic phase, you have mitosis, and then cytokinesis. Cyto means cells. Kinesis means to move. So the cells move apart. They become two separate daughter cells. Look here, you have one cell and another, right? So, and then each of these cells will undergo their own interphase and then they'll undergo mitotic phase again, right? So now let's start with interphase, right? G1 is our first growth phase. This is a period of active growth, right? You're going to have an extensive amount of synthesis of new organelles. You're going to have the cell growing so that it reaches its mature size for its specific type, right? So this cell will grow, let's say it's a neuron, it might start off small. Then you're going to have it go and keep growing to the size it should be, right? So at the same time as this cell is growing in size, it's also growing in its function. So it's making a lot of the regulatory proteins that it's supposed to do. So maybe a cell in your stomach will start making that acid, the hydrochloric acid that it needs. Maybe a cell in your nervous system will start making the protein channels it needs on its surface to allow ions to flow through to generate electricity. They'll start making those important proteins that are required. So again, this is an interphase, and it's the growth one part of the cycle. Okay, in interphase, we also have, following that, the S phase. Among the many preparations that a cell needs to make during interphase, one important task is to then replicate its DNA, right? So during this S phase, this synthesis phase, that's how I remember it, it's DNA synthesis, S, an exact copy of each chromosome needs to be produced, right? We have our amount of chromosomes in our each of the cells, right? And this cell, it will make a copy of each of those, right? Why does it want to do that? Because eventually it'll want to split apart, and every cell needs to have the same genetic information, right? So this actually takes a bit of a while, and it takes about half of the cell cycle to do this, okay? Now, let's focus in on S phase a bit. When a cell is not dividing, right, you have this type of, the DNA is actually not neatly put into chromosomes. You actually have what we call chromatin, right? Chrome, because it's still genetic information, chromatin is all loosey-goosey. It looks like a bunch of spaghetti in the nucleus, okay? All that DNA is unwound. So when a cell is not dividing, the DNA inside the nucleus exists as a very long, unpackaged fiber, not visible with a light microscope, and it's referred to as chromatin, okay? When a cell prepares to divide, it's ready for division. If it were to divide with this crazy spaghetti, it wouldn't go good, right? The noodles would go everywhere. Instead, what you want, when you're ready to divide, right, DNA is tightly packed and condensed, not into chromatin, but instead into what we call chromosomes, right? Look at these nice chromosomes. All the spaghetti is packed into nice little chromosomes, right? And now the cell is ready to go, undergo its division, right? And chromosomes are actually more easily moved around, and the cell could better organize, okay, you're going to go here, you're going to go here, and it really wants to get the right genetic information to each cell. Imagine if a chromosome was missing in a cell. A whole bunch of genes that are crucial for its function would not move on to the next cell, and that wouldn't be good. We lose genetic information, our cells would probably die, right? So if you put them like this, the cell can better organize where each chromosome, part of the chromosome needs to go, and they'll split them very nicely. We'll talk about that in a minute. And secondly, you could see these under a microscope. Like you could take a microscope, a light microscope, and you really see these chromosomes very nicely organized into this, these two little sticks over here, okay? And they're attached at a certain point. So what does chromosome number have to do with this, right? Let's talk about our chromosomes. Why chromosomes? Just because it's easier to talk about them, because they're nicely packed and visible, and they're actually, they're not in this spaghetti noodleness. So it's easier to talk about your chromosomes. Humans have 23 different kinds of chromosomes. Where do these come from? One complete set of these come from your mother. 23 of these come from your mother. Look at the egg that fertilized you, right? You have all these 23 chromosomes. One other complete set comes from your father, right? Here are the 23 other ones from your father. And when a sperm and an egg come together and form the baby's cell, right? This is like your specific cells have a set of 23 from your mom and a set of 23 from your dad. And so in the end, each of your cells, right, your hair cell, your neurons, they each have this set of chromosomes, right, that kind of match up, and we'll talk about that later on. So this gives us a grand total of 46 individual chromosomes. But remember that there are 23 different kinds. So chromosome 1 from your mom and chromosome 2 from your dad, right? Those are one kind, but there's two of them, one from your mom, one from your dad. And we'll talk about what that means later on. Now, on the other hand, if you have an eggs or sperm, right, look at the number of chromosomes. They actually have half the number, right? They don't have 46 individual chromosomes. They have 23. They have half the number because they're going and fusing together with an egg, and dad gives one half and mom gives the other half to make these cells. So all the neurons, the muscles, and everything, those somatic cells all have 46 individual chromosomes, right? Which are all 23 pairs, right? And then the egg and the sperm only have half of that complement because they're providing it onto the offspring. Now, we call this a karyotype when we look at all the chromosomes. We profile our chromosomes. This chromosomal profile is arranged by number and by size in humans, and we characterize them into two groups. The first group are called the autosomes. There are 22 pairs of chromosomes found in both males and females. So over here, this is a typical human, right? And chromosome 1 has a copy from mom and a copy from dad. Chromosome 2 has a copy from mom, a copy from dad, on and on and on and on. So all of these 22 purple ones, right? One comes from the mother, one comes from the father. They're called autosomes, automatically given, right? Sex chromosomes, there's one pair of chromosomes that is unique to your sex, right? So over here you have this X and Y, right? That's the 23rd pair. That's that 23rd chromosome. Remember I said there was 23 different kinds of chromosomes? That's that 23rd one. And you'll either have an XX or you will have an XY and be this biological male or biological female, right? Now, for those autosomes, as I said, one complete set of 23 chromosomes are from your mom and one complete set of 23 from your dad, right? And then you have the sex chromosomes. That's a bit of a different story, but we won't focus too much on that. Now, a karyotype, this is what I was saying about. So you have those 22 chromosomes, right? And if you're a male, you have this XY chromosome. And if you're a female, you have this XX chromosome. Okay, it should say X over here. And so sex chromosomes, right? One pair of your chromosomes is very unique to your whatever biological sex you have. Now, let's go back to our thinking. So we talked a bit about chromosomes and chromatin. Chromatin was the loose spaghetti of your DNA in the nucleus. And when it wants to replicate and make two cells, right? Then that chromatin goes and bundles back up into a chromosome, right? So that it's ready to undergo division. So remember that in S phase, we want to make a new copy of our genetic material, all our DNA, which means all of our chromosomes, right, need to be replicated. The process of duplication of a DNA molecule is called DNA replication, right? We are replicating the DNA, all right? So each parental strand, this is called the parent strand, okay, serves as a template for the synthesis of a daughter strand. Here we see two parental DNA strands. So in a cell, it's not like it's from two different parents. You call this the parent DNA, right? So it has a double wound. That's the parent cell, right? So let's say I have a skin cell, right? It has this DNA with this wound up double helix, right? These are the parental strands we call. These parental strands, right, when they undergo division, there's this replication fork that splits up the DNA, kind of like transcription in a way. I don't want to say it that way, but it's a bit like that. And when you duplicate your DNA so that you could send it to one cell and another, you're going to have one of those strands, right, that's going to be parental, and the other strand needs to be synthesized. That's why it's called DNA synthesis or DNA replication. You need to replicate one part of the DNA so that you have the right copy, right? There's like two different strands intertwined. So look at here how in this replicated DNA, we have one parental strand and one daughter strand, right? Just like when we call it the daughter cell. Daughter just means a new one, right? So to that generation. So here you have on this side, one parental dark blue strand and one light blue daughter strand. So we actually call this DNA replication, right? This is not transcription or translation. We're not making a product, a protein. We are going and replicating our DNA. We're synthesizing this, we're making a carbon copy of our DNA, right? So that we could pass it on to the next cell. We're not going to express a protein or that. We're taking our DNA and making an exact copy so that we can then split it into two cells. So DNA, this S phase, when we replicate, right, we call it semi-conservative. Because after replication, each new DNA molecule will consist of, here you have your parental duplex, right? You're going to end up having one parental strand per helix, right? You see the blue one. And then you have one newly synthesized daughter strand, as I said. This is the daughter strand number 1. That belongs to one of them. And you have the daughter strand number 2. So it's semi-conserved because you keep one of the parent strands and the other one you make a daughter strand. And this DNA will go on to one cell. This DNA will go on to another cell when there is a division, right? And the DNA will be exactly the same in each of the two cells, daughter cells, as it was in the parent, right? Now, a bit of weird terminology is about chromosomes. So we have different chromosomes, right? Chromosome 1, 2, all the way up to 22. And then we have the 23rd pair is our sex chromosome. So our chromosome, let's say chromosome 1, right, is a pair of homologous chromosomes. So basically, you have two chromosome 1s. You have two chromosome 2s. You have two chromosome 3s, and on and on. Let's go back and look at our little chart over here, look at chromosome 1. You had a maternal copy and a paternal one, right? And all of these have that. So what happens, right, is these are called homologous chromosomes, right? Homo means the same. Homologous means the same chromosome, right? Except for a few little differences, okay? So a pair of homologous chromosomes is a set of one maternal and one paternal chromosome that pair up with each other inside the cell when they replicate, right? When there's replication, these two will go and pair together, right? They'll bundle up their chromatin into chromosomes and then they'll pair together. And that's very important, okay? So these homologous chromosomes, one of them over here, right? Let's say this is chromosome 1. You have chromosome 1 from your mom, 1 from your dad, right? So over here, right, when you have these copied chromosomes, right, during this replication, when you go and split up your DNA to make two separate cells, you're actually gonna replicate during S phase, this DNA synthesis we kept talking about. Over here, when we said the DNA splits up and makes a new daughter strand, right? You're actually, here you see these sticks in the homologous chromosomes, but it's all actually a bunch of DNA all wound up from mom and one from dad, right? During S phase, you're gonna go make a carbon copy of this one from mom, and you're gonna go make a carbon copy of the one from dad, exactly the same, right? This is what we call sister chromatids, right? Chromatids, right, are exact copies of a chromosome. So these two, this is like one chromosome with two sister chromatids, right? This is another chromosome, right, with two sister chromatids. It's just that there's a copy in it, right? So these copied chromosomes are referred to as sister chromatids, right? Both contain the same genetic information, and they're physically attached to each other by what we call centromeres, right? Do you see this little dot in the middle? That's called a centromere, and that's where the two identical chromosome, the chromatids, are brought together. So there's a lot of lingo. First of all, there was chromatin. Chromatin was unwound DNA, all loose like spaghetti in the nucleus. A chromosome condenses into this straight stick, right? When it's ready to go and divide the cell and give each its DNA. There are homologous chromosomes. Chromosomes are those compacted forms, right? So you have one chromosome, one of those homologs are from mom, the other is from dad, right? These are homologous. Homo means the same chromosomes, except that one's from mom, one's from dad. And here too, we call this one chromosome from mom, one chromosome from dad. The only difference is they're replicated. So we say they have these sister chromatids, right? Sister chromatids. Now, so these copied chromosomes, these sister chromatids, right? They're now going to be able to be split up nicely to go to each cell. We made a copy of the DNA that needs to be given to the next cell. So now we're going to go on to the next part. Let's clarify our vocabulary of chromosome, chromatid, chromatin. Okay, there's a lot going on. So the chromosome, remember, it's a linear piece of condensed DNA. You don't see it here because it's just in color, but there's a bunch of spirals of spaghetti all here, all compacted, right? Chromosomes are numbered, right? This is chromosome 1 from mom, chromosome 1 from dad. Differently numbered chromosomes contain different genes. Chromosome 1 might have genes for the proteins in your eyeball and your skin, right? Chromosome 2 might have some genes for your stomach and your intestine. Chromosome 3 might have information for your brain and your spinal cord, right? I'm making all these up, but each of them has different information. Chromosome 1 from your mom tells you what to do for, let's say, your skin and your eyeball, right? It has that genetic information to make the proteins, remember? And then the other chromosome 1, right, is from dad and still has the information for your skin and your eyeballs, right? So they have the same information if they're the same number, okay? Homologous chromosomes that have the same number, right? Chromosome 1 from dad and chromosome 2, over here, I made a mistake. Chromosome 1 from dad and chromosome 1, this should be a 1 here, from mom, right? They have the same genes, but different, what we call alleles, so different variations. So again, I talked about the eyeball, right? Maybe there's a little gene here for the eye pigment, right? Well, on dad's chromosome, over here will also be the same information for eye pigment on chromosome 1. The only difference is mom might have the blue-eyed gene and dad might have the brown-eyed gene, right? So they're the same gene, they're just a variation, which we call an allele. Don't worry about the word allele, okay? But it's a different variation. It's the same gene. It's the eye color gene, okay? So over here I ask you, how many different chromosomes do you see in the doodle, right? So think about, again, what we say about chromosomes. Here, right, the terminology, we have, this is one chromosome, right? When we went all the way back, let's go see here, right? So we said that we have 23 different kinds of chromosomes, and this gives us a grand total of 46 individual chromosomes, right? So if I come to my little drawing here, this is one chromosome, right? This is another chromosome. So these are going to be two chromosomes, 4, 6, 8, right? On and on and on. 2, 4, 6, 8, 10, right? So over here we have 10. Here we have 20. We have all the way to, we'll have 40, right? So we're going to take this number that we have, 23 chromosomes, and we're going to multiply it by 2, because for each of them, we have 2 copies, right? So in the answer to this, how many different chromosomes do you see? You could say, I see, over here, I see 23 pairs of chromosomes, right? 23 pairs of chromosomes means 46 individual chromosomes, okay? Chromatids, right? Remember, during S phase of interphase, chromosomes, right? The linear pieces of DNA, they're duplicated, right? Over here, I had them alone, which is normally how we see it. After we go through S phase, then we get a doubling of them, right? This is when they're getting ready to go and replicate and go and divide the cell. So after S phase, chromosome 1 from dad is composed of 2 identical sister chromatids, right? So here I have 1 chromosome made of 2 sister chromatids. Here I have 1 chromosome made of 2 sister chromatids. In total here I have 2 chromosomes, each having 2 sister chromatids, okay? This is still chromosome 1 from dad. This is still chromosome 1 from mom, okay? All right, so again, how many different chromosomes do you see? I see 2 chromosomes, right? And then I could say I see a pair of chromosomes, right? 1 from mom, 1 from dad. So that is going to be our answer. How many sister chromatids, though? I see 1, 2, 3, 4 sister chromatids, right? that means little sister replicated chromosomes. So this is actually a good picture. So the cell before chromosomal duplication, you have them separate. You have one sister chromatid, right? And it's going around in the nucleus doing its job. Then the cell, after duplicating, right, has one and two sister chromatids. This is still one chromosome, and this green one is still one chromosome, right? So here we see two chromosomes. And then after division, right, they get split apart, and one cell gets a blue, one gets a green, the other one gets a blue, the other gets a green. Do you see how these daughter cells have the exact information as the cell that was at the beginning, before chromosomal duplication, right? Okay. So now the last thing to make sure we understand is chromatin and chromosomes. During DNA replication, chromatin condenses into chromosomal form. Look at this chromatin. It's a big block of spaghetti, right? When it's ready to go and divide the cell, it goes into these thicker chromosomes that are more easily placed around, okay? I know it may not look like it, but you actually have gummy worms over here that you can more easily go in place compared to this spaghetti. So the chromatin is spaghetti, then it goes into chromosomes. You see that very well in this picture where they're nice and neat. Okay. Now, let's make sure we have our good count. So before S phase, right, when we just have our 1, 2, all the way to 46 chromosomes, right? We have one chromosome from mom, chromosome 1 from mom, chromosome 1 from dad. That's 2. Chromosome 2 from mom, chromosome 2 from dad. That's 4, right? So 2 times these 23 will give me 46, okay? X and Y is like our 23rd pair of chromosomes. How many sister chromatids do we have? None, right? None of them are sister chromatids, okay? Then we undergo our S phase, and after S phase, how many, you see that each of these went and duplicated. 1, 2, 1, 2, 1, 2, 1, 2, right? Each of them have a pair of, and 1 and 2 is identical, right? How many chromosomes do we have? We still have 46. This is one chromosome from mom, right? That has 2 sister chromatids. This is one chromosome from dad with 2 sister chromatids, and on and on and on. So we still have 46 chromosomes. We just have over here the duplication, right? So over here, the number of sister chromatids, right? The number of pairs is going to be 46. We have 46 pairs of sister chromatids. But then the number of individual chromatids that we have is going to be 46 times 2, right? So let me show you what I mean. Here you have 1 sister chromatid, 2 sister chromatids, 3 sister chromatids, 4 sister chromatids, right? There's 4 per chromosome. We have 23 chromosomes. 4 times this 23 will give us our 92. But if you ask the number of sister pairs of chromatids, right, then you'd have, this is 1 pair of sister chromatids, 2 pairs of sister chromatids, 3, 4, on and on and on, right? That adds up to our number of chromosomes that we have, okay? So make sure you understand this terminology. That was DNA synthesis. So interphase. What is interphase? It's that first we have a growth 1. We just do the stuff that this cell was supposed to do, right? It was designed to secrete hydrochloric acid in the stomach. It was designed to conduct electricity in a neuron. And then in S phase, then the cell starts at the same time making a copy of its DNA, right? So it starts going through this process to make a copy of each of those separate chromosomes, right? Once each chromosome has a separate copy, now we move on to G2 phase, right? Growth 2. In growth 2, this is the final stage of interphase when that second growth phase occurs, right? And the cell increases in size slightly, right? And what this cell will do is it'll prepare for the mitotic phase that's to come. And what will it do? It'll duplicate its centrosomes. Do you see this over here, these centrosomes? There was this pair of centrioles, right? It duplicates it, right? The chromatin, look at it here, is duplicated, right? And so now really we're in our G2 phase. We have our duplicated chromatin. We have our duplicated centrosomes, right? And so these centrosomes, remember, are these two centrioles, and these centrosomes form the microtubules, right? You have these little microtubules, which during cell division are called spindle fibers, and they're going to pull that DNA that got, the DNA that got duplicated, and they'll pull them to each pole of the cell so that you can then, each cell will have its own DNA. Right? Again, the whole point of this cell division is we need to get DNA duplicated and into each of the daughter cells. So after G2, right, our interphase is done. We grew, we synthesized our DNA, and then we grew again. Then we actually have to undergo mitosis. So we have to go and have our mitotic phase. So mitosis, right? Now that we duplicated our chromosomes, we're ready to go and actually equally distribute that DNA into each of the new daughter cells. And it's really broken down into 5 stages. You can take a look at this little video. It's quite helpful. The first phase is called prophase, okay? Then you have this prometaphase, metaphase, anaphase, and telophase, okay? So we're going to go through each of these separately. Now, just put yourself in context that remember in the G2 phase, we replicated our centrosomes, and on top of it, we have our DNA that's duplicated. So let's start with prophase, okay? Chromatin will go and compact into chromosomes, right? So 2 identical sister chromatids per chromosome, they will start to go and compact into their chromosomes, okay? They're not going to be chromatin anymore. The nucleus remains intact, but the nucleolus starts to degrade. Duplicated chromosomes separate and move around the nucleus, right? The duplicated centrosomes separate and move around the nucleus. So you see these centrosomes, they start moving around, right? And then microtubule fibers extend between the centrosomes to form the mitotic spindle. They start forming their little microtubules to get ready, okay? That's called prophase. Like the prologue, right, is always at the beginning. Metaphase is next. So this is like the kind of to the metaphase. The nuclear membrane breaks down. Do you remember this nuclear envelope, right? The membrane will go and break down now. We no longer need to protect our DNA, right? We break down our nuclear membrane, and the centrosomes, right? These little centrosomes here are now positioned at opposite ends of the cell, ready to go, okay? Some microtubules will attach to a protein called the kinetochore. In the middle of this little chromosome is the centromere region, right? You see it over here. The centromere is where this duplicated chromosome connects to, right? And you have each sister chromatid on each side of the centromere. And you have these little kinetochore proteins where the microtubules will go and attach to, right? So some microtubules attach to a protein over here called the kinetochore. And what they're going to do is push these chromosomes to the middle, the equator of the cell. Okay, so some of them will start to attach and push them to the middle of the cell. That's prometaphase. It's like getting ready for metaphase. Chromosomes are now all attached to these spindle fibers. Remember, the centrosomes that send out these microtubule spindle fibers, they connect to each of the little kinetochores, and they align the chromosomes in the midline of the cell. This is called the metaphase plate, right? So over here in the metaphase plate, you have the spindles that pushed all of the chromosomes to the middle. And for each chromosome, the kinetochores of the sister chromatids, they're attached to the microtubules, right? So each of these chromosomes needs to be attached to these spindle fibers, the microtubules that are there, okay? That was metaphase. Anaphase, right? Now we're actually splitting these copies. Remember, this is one sister chromatid, another sister chromatid, split now into two and become separated, right? These are copies. It's good because this cell that will be formed over here will have this copy, and this cell that will form here will have the other carbon copy, right? So they're each going to have a copy of what the genetic material was, right? Each of those individual 46 chromosomes will be sent to each side, right? One sister chromatid from each of those chromosomes. So during anaphase, the centromeres break and the sister chromatids are separated, right? They are now considered daughter chromosomes, right? And they're not chromatids anymore. They're not stuck together, right? They're new daughter chromosomes. And the chromosomes are pulled to opposite ends of the cell using the microtubule tracts, right? So the cells will then elongate a bit. And at the end of anaphase, each pole of the cell has a complete set of the chromosomes, right? 46 individual on one side and 46 on the other. I know here there's only 4. It's so that we could more easily understand what's going on. The last phase is the telophase, right? Telo means the end of. So one copy of each chromosome is now present at the opposite ends, right, of the cell. And the chromosome starts to loosen and become less coiled. Remember, that's called chromatin. They start kind of getting looser, right, into chromatin form. Microtubules, right, the spindle fibers disappear. They go away. And the nuclear membrane starts to form around the DNA chromosomes, okay? So telophase marks the end of mitosis, even if cell division is not entirely complete. Okay, we have one last little thing we need to do. So you see very nice that you have this, all of this wall starting to be formed around the DNA. The DNA will actually start to loosen. Remember, we have 46 chromosomes on one side, the same 46 on the other that were duplicated. The last part, right, is cytokinesis. Cyto means cell, kinesis means move away. So these two cells, right, need to now move away from each other and not get back together, right? So this is cytokinesis. So this begins at the end of anaphase. Here there was remember we had anaphase, we had telophase, and then in the mitosis we had anaphase, telophase, and then we move into cytokinesis. This describes the division of cytoplasm, this is the inside of the cell, via the gradual pinching inwards along the midline, forming a cleave furrow. What does this mean? It means the cells split apart. That's all that it means. The cytoplasm, the soup of the cell, splits into all that soup gets divided in the two cells, and you make a new membrane that pinches off, right? So two new daughter cells are formed with identical DNA and equal cytoplasm volume. Look at this over here, right? The cells, this is like the end of telophase, right? And then the cells pinch off here. That's cytokinesis. This here describes cytokinesis. It's when the cell, right, goes and pinches off and becomes two separate cells. Yes, we have now our two nuclei that are good, but now we need to pinch off the middle, and this is called the cleavage furrow, right? This is where the pinching off of the cell goes on. So the two new daughter cells are formed with identical DNA and equal cytoplasm volume, right? The cells can enter now, this cell over here could enter G1 of interphase and go through it the same loop again, right? And the other cell could also go through its own interphase, right? Do you see the main thing here to note is these are identical cells. They're twins, right? They're the same genetic information. This is not meiosis. This is mitosis, right? Where the cells are genetically the same, like your skin cells. They're going to replicate the same type of cell, right? Okay, that was a lot. So let's go through a bit of some review. The number of chromosomes, right? Assuming the cell below is a human cell with 23 pairs of chromosomes. Here we only saw examples with like 4 chromosomes, right? But actually we have 23 of these, right? Here you see 2 chromosomes, but we have actually 23 of these. Let's go count what goes on, and note that the number of chromosomes does not change, but the chromatid number does, right? So individual chromosomes, we have 1 from mom, 1 from dad, 2 from mom, 2 from dad, 3 from mom, 3 from dad. So we have in total, right, we have 23 pairs of chromosomes, each different genes on them, but 23 of them are from mom, 23 of them are from dad, so that makes a total of 46 chromosomes. When we replicate our DNA in S phase, we still have, this is still considered one chromosome, right? So this is 1 and 2, right? This was 1 and 2. This was now 1 and 2. So we still have 46 chromosomes. Same thing in mitosis, 46. And same thing when the cell separates, each of these cells has 46 chromosomes, right? What changes is the chromatid number. Here, I technically have, right, 46 of these little chromatids that are there. Then they duplicate. The same chromatid is going to be, you're going to have a duplicated carbon copy, right? Remember that this is between mom and dad, right? The red one and the blue one, they're the same chromosome, which means they have the same genes on them, but they have different variations in the genes. Remember, it's like the blue eye, the brown eye. These, between these two red sister chromatids, they're exactly the same. They're mom's brown eye, and on the other one, there's mom's brown eye. There's mom's, skin protein, elastin, and there's mom's skin elastin protein, right? The genes are the same and identical. The versions are identical. Whereas between red and blue, right, mom and dad, the versions are different. Maybe blue eyes and brown eyes, okay? So the chromatids, here there's 46, right? Then they duplicate. And then there's 46 times 2, there's 92. Here there's 92, right, during mitosis. And then we're back to 46 once the cells split apart. So let's go through this one last way. During cell division, right, examine the nuclear envelope, the position of the centrosomes and chromosomes, and the shape of those, right? So let's start at G2, right? After growth 1 and after synthesis, you have replicated chromatin, right? And you see that over here. You see that you start having that replicated chromatin, and you have the green, the centrosomes, right, that are starting to spread a little bit. Then in prophase, right, you end up having your early spindles, right, starting to pull out. You really see those spindles starting to form. And you have your nucleus over here in blue. The blue is staining for DNA. Look at the spaghetti, right? Now the spaghetti is starting to compact and condense. And finally, in prometaphase, we really are condensed, right? Look at the chromosomes. You could count them easier because they're nice and condensed. And look at the little centrosomes are on each side during prometaphase. They're positioning themselves on each side and they're sending out their spindles, right? During metaphase, right, remember the little spindles are going and attaching to the kinetochore in the centrosome of the chromosome, right? So they send that information to each side. During anaphase, they're splitting apart the pairs and they're actually pulling it towards each end, right? These blue ones are going to be pulled towards this end. These blue ones will be pulled towards this end. And at the same time, we start the cleavage cytokinesis process, right? So here you see very nicely that you're pulling away all of those to each of the ends, right? And cytokinesis is starting to happen. You see this cleavage furrow, right? Where you're starting to have this cut in the middle of the cell so that they can form their own cell. So take a look at this over here. This will help you to figure out what phase of mitosis, this is the mitotic phase, right? So the prophase, anaphase, try to place these all in the order that they should be and what phase of mitosis it is, right? This will help you a bit study the different phases of mitosis. I'm going to play this little video because I think it is super helpful and useful to look at it in all together, right? So let's watch this and then we'll go on to the next thing.
This soccer player's knee will be as good as new in a few weeks, thanks to mitosis, a type of cell division that generates new cells for growth and repair. Let's move into a cell to witness the events of mitosis. Before a cell can divide, it must first duplicate the chromosomes stored in its nucleus. During chromosome duplication, several bubbles open up along the chromosome. Each bubble grows until it merges with an adjacent bubble. Each chromosome now consists of two identical copies called sister chromatids. Getting closer, we see that each sister chromatid consists of DNA wound around small proteins called histones. The sister chromatids begin to coil into tight helical fibers. Outside the nucleus, centrosomes that duplicated earlier move away from each other to opposite sides of the cell. Microtubules extend from the centrosomes, forming the mitotic spindle. Back in the nucleus, the DNA forms loops, becoming more compacted. These structures fold back on themselves, eventually condensing into a shorter and thicker chromosome consisting of two sister chromatids. As the chromosomes continue to condense, the nuclear envelope breaks up. The array of spindle microtubules is now extensive, and the chromosomes are fully condensed. Spindle fibers from each pole attach to protein structures located at the centromere of each sister chromatid. As the chromosomes are bound by spindle fibers from opposite poles, they move first one way and then another. The counteracting forces of the spindle eventually cause all the chromosomes to end up at the center of the cell, as if arranged on an imaginary plate. The sister chromatids are released from each other, each becoming a full-fledged chromosome. They are moved toward opposite poles of the cell, pulled along the spindle fibers attached to them. At the same time, overlapping spindle fibers that are not attached to chromosomes continue to lengthen, pushing the poles farther apart. Once the chromosomes arrive at their destination, they become less condensed. Two new nuclear envelopes form, completing mitosis, the division of one nucleus into two genetically identical daughter nuclei. The cytoplasm divides by the process of cytokinesis, forming two separate daughter cells. In your body, millions of cells divide every second, providing new cells for growth and for repair of damaged cells.
All right. So you saw all of that kind of mitosis, right? The cell division from really interphase all the way to the mitotic phase. So now we went through cell division. That was the hardest part. Now I'm just gonna add a few more layers about what cells do and why abnormalities in cancer. So first of all, we're going to now talk about exiting the cell cycle. It's not always that cells are continuously cycling in this interphase, mitotic phase, on and on and on, right? They could go into G0, right? Growth 0. They're not growing, right? So the cell cycle length is unique to every cell type and organism, first of all. The time it takes for cells to pass through the cell cycle depends on the type of cell and the organism's stage of development, right? So basically, the type of cell, right, maybe a cell like a stomach cell gets damaged easier, so it goes through a lot more cycles quicker, right? Maybe a cell like that doesn't get damaged as often doesn't need to do so. So actively dividing cells in some human tissues, such as the intestine, the skin, they need to divide quickly and frequently. These cells complete the cycle in about 12 hours, right? They really need to go and keep making new cells. They get damaged all the time. Think of our skin cells always exposed, right? Same thing with the intestine. There's always food brushing through and ripping it off, right? Most other actively dividing cells in your body take about 24 hours to complete the cell cycle. Unicellular organisms like yeast can complete an entire cycle in just 90 minutes, right? So it really depends on what you're talking about, if the tissue needs more cells more often, right? And so we're going to talk about this G0 phase. Not all cells in your body actively participate in the cell cycle, since not all tissues require rapid replenishing of the cells. Yes, your skin cells do. Yes, your intestinal cells do, but not other types of cells, and we'll talk about which ones. Many cells pause in the cell cycle somewhere between the M phase and the S phase, and they exit it and they enter this G0 phase, okay? And it's distinguished from growth 1 phase by the absence of the preparation of S phase. So you're not going to have an S phase here. You're going to wait, and then eventually you'll return and enter S phase. Okay, you're not going to be duplicating your DNA, therefore you won't be continuing for division. Although these cells have left the cell cycle, they're still active in other ways, right? They still undergo transcription, translation, gene expression, right? They still go and do their functions in the tissues. So an example of this is neurons, right? Cells of the eye lens and nerve cells are perpetually in G0, and they're therefore non-dividing cells. Neurons do not divide. That's why when there's a stroke or there's some kind of neurological disease, we get scared because neurons don't replicate themselves for the most part. And so neurons don't make new neurons, and therefore when that region's gone, it's gone, right? Liver cells, called hepatocytes, they remain out of the G0 phase for about a year, right? So they carry out metabolism and detoxification before re-entering the cell cycle. So they do a lot, they actually have a pretty long lifespan before they go and replicate again. So these are examples of, here G0, neurons are always in G0, and liver cells for a year, they're in this G0 phase, okay? So the cell cycle length is unique to every cell type and organism. The time it takes, right, to pass through the cell cycle depends on the type of cell and the organism's stage of development. Neurons, for instance, do divide in the beginning in embryo, but then later on in adulthood, they don't, right? Actively dividing cells in some tissues, such as intestines and skin, we said that those cells, they cycle a lot, right? Those 12 hours, whereas the ones that were actively dividing in the body, they were just going about 24, okay? So we saw this already. Regulating the cell cycle, okay? So controlling the cell cycle phases, right? This is going to be important. So we talked about the cycle, but now not every cell divides, right? And cells that do divide must do so in a very controlled order and regulated manner. If cells divide when they shouldn't, a lot of trouble occurs, and this is why the cell cycle is very tightly controlled. There's 3 factors when a cell should divide properly, okay? So cell division needs to occur at the right time. During injury, just enough new cells must be produced to return to its optimal function, just like a scratch on your skin, right? Or some kind of injury, you need to make the right amount of cells to re-epithelialize your skin. So that's time. Cell division occurs in specific locations. Only properly placed cells should be dividing. So a detached cell that wanders into new tissue won't be dividing and establishing itself there. What if a liver cell detached and went to your brain, right? You don't want a liver cell in your brain. We're going to talk about tumors, and this is what happens. Cells go to regions they're not supposed to, and they make tumors, right? And then they spread to other regions, and on and on and on. Cell division also needs to occur in healthy cells. Abnormal cells or old cells that aren't doing their proper function anymore should not undergo cell division, and they should be killed. We'll see this brutal effect of the cell. Now, there's 3 very important regulatory mechanisms that control cell division. We're going to go through each of these. The first one, right? The first 2 are pretty not so complex. The third one, we'll talk about the checkpoints, okay? So the first one is receptors, right? They monitor the surface molecules on neighboring cells, right? So receptors monitor the surface molecules on other cells to monitor cell density in a tissue. And this leads to what we call the big word, density-dependent inhibition of growth. What does that mean? That means that cells will move and divide only if space permits. Let's look at this example here. Cells anchored to a dish surface and they divide. Do you see these cells here? If you put them in this dish, they'll keep dividing. When cells have formed a complete single layer, they stop dividing. That's called density-dependent inhibition. This is dense enough. I have one nice layer, right? And I don't need to keep growing. So when the density is optimal, then there's signals between them that say, whoa, we're done. If you go in and you scrape away just a little piece off of the plate, cells will actually divide just to fill in that gap, right? So the cell density is limited to a single layer by contact with neighboring cells and available nutrients. In cancer over here, cancer cells usually continue to divide well beyond that single layer. Look at them, they're stacking, right? So they form a clump of overlapping cells. That's what a tumor is usually. And they don't exhibit this anchorage dependent or density dependent inhibition. They stack on each other and create this huge layering, right? So cells, so density means the right amount of, when they're the right amount of cells, they will inhibit the growth of new ones, right? So cells will move and divide only if space permits. For example, cells can sense density levels in a tissue and will cease division if density is too great. They'll actually go into G0 and pause. These cells will only re-enter the cell cycle if cell density drops. Okay? So that was for density-dependent growth inhibition. Now we're going to talk about anchoring proteins that hold cells in place within their tissue and prevent cells from establishing themselves in other tissues if they're dislodged, right? This is called cell adhesion. Cells have adhesion to the basement membrane usually and to themselves, right? In healthy tissues, cells remain locked in place via anchoring proteins. Here you see the yellow proteins anchor them to their connective membrane, connective tissue underneath, right? If anchoring is lost, the cell is either fixed and re-brought, it's either killed, or it's told to commit a cell suicide. And we'll talk about this apoptosis, okay? So over here, right, normal cells that are all tightly packed at the region that they're supposed to be, if one of them kind of separates from this region, right, and ends up going into the bloodstream, right away the cell will go and kill it, right, or tell it to go and die. If this tissue, right, does not, one of them gets separated, right, but instead it doesn't get killed right away, right, and it enters the bloodstream, this is not good. This is how cancers and tumors and malignancies, metastasis happens, right? So you need adhesion to really lock you in place. If you're not adhered, there's some signals that might kill your cells, right? We'll talk about them. Exceptions do exist, such as cells of the circulatory and lymphatic system. All your blood cells, all your white blood cells, red blood cells, could leave the tissue and they're not going to die, okay? A tumor, right? Remember, a tumor, if those cancer cells invade neighboring tissues and go in the blood vessels or the lymph nodes, right, then they metastasize, right? This is why we have adhesion signals to see if a cell should go and be killed or not, right? So the last one is these checkpoints. So DNA and cellular function. What other regulatory mechanisms do we have? We had this density one, adhesion, and then we have these cellular checkpoints where regulatory proteins during the cell cycle will go and kill the cell if it's not behaving. So these are called cyclical operating set of compounds and proteins. And they will trigger, right, and coordinate key events in the cell cycle. They're these little green light, red lights. Well, you can't go, you're done, you're not working well, right? At specific times, the cell checks itself to make an assessment if everything is okay. First checkpoint is called, checkpoint number 1 is the G1 checkpoint, right? You see this little wall here. There's a little stop to see if everything's good. In human cells, the G1 checkpoint, this restriction point appears to be the most important. If a cell receives a go-ahead signal at G1, then it'll go through S phase, G2, and the M phases, right? So it'll go through mitosis and divide. If it does not, the cell goes into G0, goes to sleep for a bit. Okay? So the checkpoints, they monitor different things. That checkpoint number one, well, first of all, I could put something into sleep, right? Or I could say, is there a DNA damage, right? Here you could say, is something not working where I shouldn't go duplicate my DNA? At checkpoint number two, after you've went through S phase and duplicated your DNA, after growth two, right, you're ready for mitosis, you can say, has all of that DNA been replicated? What happens if you didn't replicate one of them by accident, right? You can't go ahead and split your cell. It also asks, are there any parts of the DNA that are incompletely replicated, right? If there's a little mistake, then it needs to go and fix that. And then checkpoint number three, right, in the M phase, they're asking, are my chromosomes, before I go split up this cell, right, are my chromosomes properly aligned? Are the kinetochores attached to the spindle fibers? If not all of the kinetochores are attached, then it stops and it doesn't go through this whole mitosis, okay? So these checkpoints are controlled by special regulatory proteins. One of the most important regulatory proteins are called P53, and it's represented by this green little bean over here. P53 is called a transcription factor, right? That means it transcribes certain things, and it's also known to be a tumor suppressor protein. It suppresses, right, tumors and doesn't allow tumors to go on. So over here, let me show you what's going on. You have this P53 protein found in the nucleus. When the DNA is damaged, right, let's say there's a damage to the DNA activates, right, you're going to have this DNA damage that will go and it sends signals usually, and it turns on this P53 and it phosphorylates it, okay? It activates. Phosphates, remember, activate things. So this phosphorylated P53 will go on and act to inhibit the cell cycle's G1 phase. So it will do things to stop it from going into S phase, right? Remember, S phase means duplicating the DNA. So it will go on the DNA and it will make sure that there is no going on to the S phase to duplicate. Okay, that's how these little phosphorylated P53 regulatory proteins help in stopping. So remember, look at where it is. It's in G1, right? So this is the little block that's over here. The G1 checkpoint is what we're looking at with this P53. P53 has many anti-cancer mechanisms. It can pause the cell cycle by putting it into G0. It can initiate DNA repair. It could send in some workers to go fix wherever that DNA is damaged. And if the DNA cannot be repaired, it will induce cell death, or what we call apoptosis. Apoptosis is another way of saying it is the cell killing itself and saying, I can't do my proper function, so I will go and create this whole catastrophe and go and die, right? So we call it also cell suicide, that the cell will go and demolish itself. So if something is wrong with the cell, the cycle does not continue until that error is fixed, right? If the error can't be fixed, the cell undergoes apoptosis, a type of programmable cell death or suicide, right? So cells should not be copied if they're damaged, because if they continue to divide, they could impact the overall function of this tissue. So over here you see the cell checkpoint, right? Here it says all violators must fix their mistakes or face apoptosis. So they need to go through this screening, right? Let's finish off by talking about cancer. So although most cells go through cell division, without any problem, a small portion of cells could be pushed through the cell cycle with dysfunctional cellular mechanisms, right? If it's serious enough, these dysfunctional cellular mechanisms could lead to apoptosis. Good, right? It gets stopped. Please die. You're not a good cell. You have some things that are not right in your DNA. You need to go and die, right? Or more rarely, right? If there's a dysfunction that doesn't get fixed, it doesn't get killed by apoptosis, then we could get cancer, right? So we're going to look at benign tumors that grow without damaging adjacent tissues. You see, it's very contained, versus things like malignant tumors that grow and spread and negatively impact the health of the surrounding tissue. Let's talk about what cancer is, right? In very simple terms, it's uncontrolled and unregulated cell growth. And it can occur in all tissues of the body, right? It doesn't matter where. Oftentimes, it'll be in regions of the body that has high replication, right? So it's caused by changes in the DNA of a cell. And this allows for it to ignore regulatory mechanisms, right? So mutations in the DNA, probably in the regions of DNA that we talked about control the cell cycle, right? If you mutated your DNA so much that the checkpoint signalings no longer work, right? Apoptosis no longer works. Density dependent inhibition signals no longer work. And the cell adhesion signals no longer are functional, right? Then unhealthy cells dividing when and where they want will go on and wreak havoc, right? Because they no longer have these things to block them from going on to their next phase. In a normal cell, in a healthy tissue, right? Let's look at what happens, right? In a normal cell with healthy tissue, we're going to see why cancers are relatively rare. So let's say one little cell, right, gets sick and gets these kind of little mutations, right? Well, and starts making these bad little cells on the side. Different types of defense mechanisms will go and kill these bad guys before it could even keep going and become malignant, right? Apoptosis is triggered if a cell is too damaged and can't repair its DNA, right? So it becomes unanchored to its tissue or behaves inappropriately. So let's say one of these cells, right, gets bad, right, and has a mutation in it. It will go and it will commit apoptosis, right, and undergo the cell suicide. In a normal cell, right, there's communication between cells through those gap junctions that we saw in the interstitial space. Neighboring cells will also trigger apoptosis, right? They'll send signals to the little neighbor and say, hey, you shouldn't be here. Please go, right? A normal cell will reply on external growth signals in order to maintain proper density and remain anchored to tissue. Remember that? So oftentimes, right, if there's something that's not kind of growing in a way that it should, then it will say, okay, you shouldn't be growing there, right? If it's not anchored down, right, then it will get killed through those apoptosis signals. So that's another way that normally we talked about cells should be going and regulating. Each individual cell has a shelf life, right? After which a normal cell stops dividing. This is called senescence. So when you are not when your life has come up, you die, and new cells come in, right? Cells have built-in expiry dates, and these likely evolved in order to avoid long-living cells that could accumulate some damage, right? So when you're done, you go on to cell heaven, you have your little funeral, and you're done, right? This is good. We want this to happen. We don't want you to stick around all the time and gain mutations and abilities to become cancerous. In a cancerous cell, what happens is that DNA is often damaged. And when this damage occurs in genes that code for proteins that regulate apoptosis, so a gene that, like P53, right, that regulates apoptosis, that regulates the cell cycle, that regulates density dependent growth, these are the first steps that could lead to cancer, right? There is less sensitivity to apoptosis signals, so unhealthy cells continue to divide and could even become malignant, right? So the cell cycle checkpoints are ignored, and so damage is ignored, right? Dependence on external growth signals is lost, so cells can grow into other cells, right? They could detach, they could metastasize and grow in new places, right? Cells ignore senescence, so a cell divides indefinitely and becomes a bit immortal, right? So cells are dysfunctional and they continue to accumulate more DNA errors and deviate more and more from those normal cells. So DNA damage, right? What causes DNA damage? There's a lot of mutations, right, that cause DNA damage. Chemical causes, asbestos and tobacco smoke. Physical causes, ionizing radiation, right? The sun's UV rays. Infectious causes like bacteria, funguses, viruses could cause this mutation. It's a self-perpetuating cycle, right? As mutated cells divide, they acquire more mutations because these mutations never get repaired, right? And on and on and on. So in a benign tumor, usual cell growth in a tissue can be benign, like polyps, moles, lipomas, right? These cells are multiplying in place and they're not moving around. Cells aren't invading other tissues, right? And cells aren't considered invasive or cancerous, and they could be removed actually. Look at this normal cell, and it makes a first mutation and it stays there, right? These are not normal cells, but they're staying in their little area. So the mutation may be in the cell cycle, right? Regulation causing the cells to unnecessarily divide. If left unrepaired, the next mutation may be more dangerous. So a malignant tumor occurs when cells multiply and invade, and this process is called metastasis, right? Other tissues causing these tissues damage. So here I have a first mutation, a second, a third, a fourth, right? And all of a sudden I become malignant, which means I could enter kind of the bloodstream and move around, right? So cells continue to invade other tissues, while tumors may induce the growth of blood vessels called angiogenesis so that it could feed itself and so that it could go around in the circulation, right? So interruption of these tissue functions leads to organ failure and death, right? This is the typical cancer we think about. This is also cancer, but it's a benign tumor. This is a malignant tumor. Okay, so here you have the takeaways from this class. Take a look at those and make sure you review these. And here are a few questions that you can think about as you're going through a bit your studying for this topic. And that's all for our cell cycle.